Hello everyone. Welcome to another lecture on micro and mol bio with Dr. Sunita Bundale. From today we will be starting a mini series on gene mutations. In today's lecture we will focus on point mutations, particularly substitution mutations, and we will also see the effects of these substitution mutations on the phenotype and the examples of it, that is missense mutations, nonsense mutations, silent mutations, and neutral mutations. So let us begin. If you like the content, please subscribe to this channel. Any sudden change occurring in the hereditary material is called as mutation. These changes may occur spontaneously during DNA replication or may be induced by environmental factors such as radiation, chemicals, or viruses. Mutations may or may not lead to changes in the observable characteristics, that is, the phenotype of an organism. They may be harmful, beneficial, or neutral. Mutations play a part in both normal and abnormal biological processes, including evolution, cancer, and the development of the immune system. So let us begin with talking about the types of gene mutations. There are a number of ways in which gene mutations are classified. They are classified on the basis of the molecular nature of the defect. They are classified on the nature of the phenotypic effect, that is, the amino acid sequence of the protein is altered or not. And they are also classified on the basis of the causative agent of the mutation. Mutations are considered very important because they are considered to be the source of all genetic variation, which further provides the raw material for evolution. They are considered to be the source of many diseases and disorders, and they are also useful for probing fundamental biological processes. So before we delve into more details, let us discuss some very common but important terminologies which we will be using in our discussion throughout. The first is mutagenesis. The process that produces a mutation is called mutagenesis. Mutagenesis that takes place without the involvement of any mutagen is called spontaneous mutagenesis, whereas if a mutagen is involved, it is called induced mutagenesis. This automatically brings us to the next term, mutagen. The agent that causes a mutation is called a mutagen. A mutagen may be a physical agent like radiations, or it may be a chemical agent like ethidium bromide. So the next question is, what is a mutant? The cell that bears a mutation is called a mutant. However, it is conventional to specify which trait the cell is a mutant for. For example, an E. coli culture which cannot synthesize lysine is called E. coli lysine mutant. In contrast, a cell without any mutation is called wild type. A mutation which converts a wild type into a mutant is called forward mutation, whereas a mutation which converts a mutant back to the wild type is called reversion or back mutation. So let us first try to understand the molecular basis of mutations. For this, we need to revise the central dogma of molecular biology, which describes the flow of genetic information within a biological system. It was first articulated by Francis Crick in 1958 and is often summarized like this. The DNA undergoes replication to produce copies of itself. It undergoes transcription by RNA polymerase to produce RNA, and the RNA, that is mRNA here, is translated to produce the proteins. The biological function of the protein is responsible for the phenotype that is expressed. A gene refers to a set of nucleotides that specifies the sequence of an RNA or protein. Changes in the coding portion of the DNA may alter the amino acid sequences of proteins. For example, the figure on your screen shows a gene with a particular sequence of nucleotides. The transcription results in the formation of an mRNA with specific sequence of codons. The mRNA is translated to give a protein with a particular sequence of amino acids. In this case, the sequence of amino acids is tyrosine, valine, glycine, threonine, valine, alanine, and histidine. A mutation occurs in the gene such that the fifth base A is replaced by C. As a result, a change occurs in mRNA sequence and the second codon GUU coding for valine is changed to GGU coding for glycine. Thus, in the protein, glycine is incorporated in the place of valine. This is the molecular basis of any mutation. Let us now talk about the types of mutations starting with point mutations. A point mutation is a genetic mutation involving mutations in a single base pair. It can be whereby a single nucleotide base is changed due to substitution, insertion, or deletion from a nucleotide sequence of an organism's genome. Point mutations have a variety of effects on the downstream protein product, consequences that are moderately predictable based upon the specifics of the mutation. Let us first discuss the substitution mutations. A substitution is a mutation in which one nucleotide is substituted by another in the DNA sequence. Such a substitution could change a codon to another one that encodes a different amino acid and causes a change in the protein product. Base pair substitution are of two types. The first type is transition, where a purine nucleotide is replaced with another purine nucleotide or a pyrimidine nucleotide is replaced with another pyrimidine nucleotide. In other words, the base in the nucleotide sequence is in the same chemical class as that of the original nucleotide. For example, AT to GC or CG to TA. The second type is transversion, when the chemical class of the base changes, that is, a purine nucleotide is replaced with a pyrimidine nucleotide or a pyrimidine nucleotide is replaced with a purine nucleotide. As a result of this transversion mutation, the mutated position in the gene may, for example, have an adenine A where it had thymine or cytosine. Comparison of the sequences of the homologous genes between species reveals a pronounced preference for transition over transversion almost tenfold, indicating that the transitions occur much more frequently than transversions. Let us now discuss the phenotypic effects of these substitution mutations. The first type is missense mutation. When a base pair substitution in DNA results in incorporation of an amino acid different from what occurs naturally in the corresponding protein, the mutation is called missense mutation. This may have a wide range of effects on the structure and function of the proteins depending on the nature of the amino acid change and its position in the protein sequence. At the molecular level, the genetic code consists of codons, three nucleotide sequences in DNA or RNA that correspond to specific amino acids. During gene expression, DNA is transcribed into messenger RNA, which is then translated into the protein by ribosomes. If a mutation occurs that changes one nucleotide within a codon, the codon may code for a different amino acid. Thus, both the codons are sense codons, but the information is misread. Hence, it is called missense mutation. One well-known example of missense mutation is the mutation that causes sickle cell anemia. This is part of the gene for beta globin, which undergoes transcription to form mRNA. Three bases each form the codons, which are translated to the globin protein. The sixth amino acid in the beta chain of hemoglobin is glutamic acid, whose codon is GAA. A substitution mutation occurs such that T is substituted by A, as can be seen on your screen. This results in the mutation of the codon from GAA to GUA. Thus, in the mutated gene, T is substituted by A, which can be seen in red color on your screen. Transcription leads to the formation of mRNA with GUA in the place of GAA. I have highlighted GUA in red color. On translation, the protein that is formed has valine in the place of glutamic acid at the sixth position. Since valine has chemical properties different from glutamic acid, this change drastically alters the properties of hemoglobin, causing red blood cells to assume a sickle shape, which can block blood flow and lead to serious health complications. Thus, a single substitution point mutation leads to a sickle cell anemia. A thinking question for you is this mutation transition or transversion? Let us move on to the next type of mutations, which are nonsense mutations. When a base pair substitution changes a sense codon that normally encodes an amino acid into a nonsense or stop codon, the mutation is called a nonsense mutation. This premature stop signal leads to early termination of translation, resulting in a truncated and usually non-functional protein. Nonsense mutation early in the mRNA sequence produces a greatly shortened and usually non-functional protein. On your screen is a gene sequence which undergoes transcription to form an mRNA. The mRNA then gets translated to form the protein with the amino acid sequence which can be seen on your screen. This is the normal functional protein. If the 10th nucleotide T is substituted by A, then in the mRNA, A is substituted by U. The mutated gene will look like this, which on transcription will give rise to an mRNA where the fourth codon is UGA instead of AGA. I have again highlighted UGA in red color. Thus, the sense codon AGA coding for arginine is converted into a stop codon UGA. This results in the process of translation coming to a halt, leading to the formation of a truncated protein which may or may not be biologically functional. Nonsense mutations early in the mRNA sequence produces a greatly shortened and usually non-functional protein. However, a nonsense mutation late in the mRNA sequence may lead to premature termination, but the protein may still be biologically functional. The next type of mutation is silent mutation. A silent mutation is a type of mutation where a base pair substitution results in a different codon which still encodes for the same amino acid. Thus, a change in the DNA sequence does not result in a change in the amino acid sequence of the protein that is produced. This happens because of the degeneracy of the genetic code, which means that there are more than one codons which code for the same amino acid. For example, This is a gene which is transcribed to give the mRNA. The mRNA is then translated to a protein with phenylalanine as an amino acid coded by triple U. In the nucleotide sequence, A is substituted by G, leading to a change in the mRNA where U is substituted by C. This is the mutated gene which on transcription forms an mRNA with UUC instead of UUU. So, the mutation changes the codon from triple U to UUC, but both triple U and UUC code for the same amino acid phenylalanine. Hence, on translation, the protein formed is such that the sequence of amino acids does not change the protein and the protein is considered to be biologically functional. There is a change in the genotype but no change in the phenotype, and such a mutation is called a silent mutation. The next type of mutation is neutral mutation. Missense mutation that changes amino acid sequence but does not alter the function of the protein is called neutral mutation. This is because the replaced amino acid is chemically similar or the affected amino acid has little influence on the protein function. For example, in the normal gene, CUU codes for leucine. A mutation changes CUU to AUU, which codes for isoleucine. Since leucine and isoleucine are similar in their chemical properties, the protein does not lose its functional function and is still biologically active. Such a mutation is called a neutral mutation. So in this lecture, we have talked about mutations, particularly point mutations, that too substitution mutation. So on your screen right now, you can see the central dogma about which I have already talked. The DNA getting transcribed into mRNA and into the protein. This is the wild type protein which is produced. Missense mutations, where the new codon encodes a different amino acid and there is a change in the amino acid sequence. We have also talked about nonsense mutations where the new codon is a stop codon and there is premature termination of translation. We have also discussed silent mutations and the new codon encodes the same amino acid and there is no change in the amino acid sequence. With this, we come to an end of this lecture. We will be meeting again in the second episode of this mini series on gene mutations, where we will be talking in detail about frame shift mutations. Till then, thank you.